Pedimos a todos que tomem seus lugares e, por favor, coloquem seus celulares no modo silencioso. O Instituto de Tecnologia em Imunobiológicos Bio-Manguinhos da Fundação Oswaldo Cruz tem o prazer de recebê-los no 8º International Symposium on Immunobiologicals. O objetivo do evento é integrar cientistas, pesquisadores e instituições de desenvolvimento e produção de biológicos, além de dar visibilidade para a produção científica, formar e qualificar profissionais e líderes de mercado e promover oportunidades de negócios e parcerias no setor de biotecnologia, estimulando um ambiente de desenvolvimento científico e tecnológico a nível nacional e internacional. Gostaríamos de agradecer aos patrocinadores e apoiadores deste evento: Sanofi, Promega, GSK,
Fiotec, Citiva,
Sykes, Cefid, Beckman Coulter, Bionovis, AstraZeneca, Fundação Getúlio Vargas e Croda. Gostaríamos de recomendar a vocês que acessem o aplicativo do evento.
You access the event application directly from our site or on the participant area that is being projected on the screen. And through this application, you will be able to make questions directly to the speakers and also participate in the research for satisfaction, and also allow that all the participants know each other and exchange information and access all the works that is registered. I remind you that this event is happening in a hybrid way, and you can follow up on the event on the application. You can also participate making your questions on person or online through this chat, and we will make that it would reach the panel's chair. The on-site people have simultaneous translation devices, and you can click on the Zoom channel on the language of your preference. These will be recorded and you can visit and revisit at all times. So starting the presentations and posters, we invite to the stage Laura Ribeiro Oliveira. She's going to present the poster subject with respiratory allergy, experience distinct adverse effects with COVID-19 vaccines.
Olá, bom dia a todos.
So, hello, good morning to you all. My name is Laura Oliveira. I am from the Federal University of Uberlândia, and I'm going to present my work, which is subjects with respiratory allergy experience distinct adverse effects with COVID-19 vaccines. So the respiratory allergy, they are triggered by inoffensive, unharmful, called aerosol allergens, and can make a severe reaction in some individuals and cause some symptoms such as rhinitis and asthma. These have happened with individuals that have the predisposition to these allergens. And with the test to verify the positivity of these allergens are the predictive tests. In the beginning of the pandemics, there was a necessity of development of many vaccines, and these vaccines were applied nationally were Pfizer with the RNA messenger technology, AstraZeneca with the retrovirus, and the Coronavac vaccine, which the technology used was of a deactivated virus. So we had worked with 305 subjects, and they were recruited through social media and by television. And these studies were carried out in the Hospital das Clínicas of Uberlândia. The majority of these individuals have the first dose of the vaccine and the second vaccine as well, and they had reported taking the booster dose, which is the third one. We also applied the rhinitis and asthma questionnaires to evaluate the symptoms of these allergies, and also the skin prick test to assess the. And also we had the ELISA for the quantification of these allergens. We also evaluated adverse reaction post-vaccination through the quantity of adverse effects that they had. Our results shown that the majority of the subjects were atopic, presenting 54.4% of the individuals, and 45.6% were non-atopic ones. And these subjects, they presented allergies, respiratory allergies such as asthma and rhinitis.
Os resultados apresentados, nós queremos analisar se a quantificação dos efeitos adversos
The quantification of adverse effects among the doses of vaccines, independently of the vaccines used, we can see on chart A that they presented having adverse reactions on the first and the third doses comparing to the second dose. On the shot B, on the chart B, we could evaluate the quantity of these effects, and you could see the AstraZeneca and Pfizer vaccines, which were shown major reactions compared to Coronavac. On the second dose, we can see a behavior on Pfizer's vaccine, which presented more effects. And comparing to AstraZeneca and Coronavac. And the third shot, there was no significant difference among the vaccines. Here we wanted to evaluate the quantity of adverse effects among the doses of the vaccine for each type of vaccine. So it's differentiated the type of vaccines. The first one is AstraZeneca, and it was able to trigger these effects in subjects that took the first doses compared to the second dose. We did not observe significant difference on the shot of Pfizer and also Coronavac. So seeing that the first dose triggered more adverse reactions, and we followed on our analysis, stratifying the atopic and non-atopic subjects, and we could observe that in atopic individuals that took the AstraZeneca vaccine, they had the more adverse reactions such as muscle pain, fever, and shivers comparing to the non-atopics that took the same kind of vaccine. These results suggest that these adverse effects can be related to atopic condition, and these can be observed, and new approaches should be carried out. Although they presented these adverse effects, they did not report anaphylaxis or respiratory allergies. So we can say that safety of COVID vaccination, they are safe especially for atopic subjects. This is my contact, and here are the researchers that works with us and also the collaborators. And I thank all the organizers. I'm very happy to be here. Thank you very much.
Obrigada, Laura. Parabéns pela sua
Thank you,
apresentação.
Laura. Congratulations on your presentation. I invite Luana Elisa Lipscher Vidal for the presentation. She's going to present the poster investigation of recombinant SARS-CoV-2 nucleocapsid protein thermal properties and its nucleic acid interaction.
Aqui muda para frente e para trás. Sim, só para frente e para trás. Obrigada. Bom dia, meu nome é Luana.
Good morning, my name is Luana. I'm from the Laboratory of Analytics Laboratory, and this work we do in a partnership with Alatea Laboratory. The COVID-19 pandemic was caused by SARS-CoV virus, and it presents the nucleocapsid and protein. It presents two conserved structural regions subdivided into N-terminal domain, NTD, which are connected by central serine and arginine here in the picture in pink. The role of N protein is not only to compose the viral particle, but also to contribute with many critical activities after cell virus invasion. And this is conditioned to the presence of DRs. The studies have suggested that CTD imitates dimerization of the N protein and also oligomers of higher forms. The work treated these with for the reduction of the nucleic acids, and we observed that after the treatment, we had the reduction of the oligomers and showing the action of the acid. With the properties of the proteins and inactive folds allow several applications, and although the control of COVID-19, they emerge continually, generating new infection cases which increase rapidly. Therefore, it is necessary the investigation for identifying the proteins for the antiviral therapy. So in this study, we investigated recombinant SARS-CoV-2 with N protein thermal properties and its nucleic acid. And in this world, we produced N protein with NA. In the comparison, we used the adrenalinase and the protein NB. These proteins were purified with these methodologies and analyzed with many biophysicals and biochemicals methodologies. The first results, we show that after the treatment, it showed a reduction of the NB protein, showing that the reduction of acid nucleic is conditioned to the reduction of the oligomers. And in addition, the ultraviolet treatment showed the treatment with abezonase was effective to reduce the acid nucleic associated with the protein N. This protein was submitted to a thermokinetic methodology, and this was submitted to 25 to 85 degrees. The protein is showed in blue, and it was very similar to the protein in its native form, so showing that the protein had the capacity of reestablish its secondary nature. And you can see in figure 2 The same occurring with protein NB. And these converted to the leveling of the proteins, we can see the comparison of the both profiles, B and NB, and showing the increase of the protein and the higher temperatures, and the same with lower temperatures. And you can see also the tertiary conditions and with the analysis of the blood. And to investigate more the thermal kinetic of protein N, it was submitted to fluorometric and also scan, where the protein presented, as we can see in figure A, two conditions showed in green from 25 to 90 degrees, and we could also observe with the re-leveling we had two transitions with similar values, showing that this protein has the capacity of reestablishing its tertiary structure, and the same was observed for protein NB. The melting temperature shows that where it was obtained through the graphics in the 300 and 330 ratio, and NA and NB protein demonstrated reversibility of denaturation process after thermokinetics. And you can see that NA had less transition, and this proved the melting tube methodology shown different results, and they have the capability of reversing the denaturation process up to 6 times. And to aid the understanding of the reversibility of the denaturation process, we could see this process where we use protein N with antibody NB, and this antibody showed different, shown in dark blue, showing an influence of nucleic acid in the antibody and protein N. The NB protein was submitted to the melting process, and it had a different behavior. And when we observe the NA renatured is different than the native protein, showing that acid nucleic allows, and that suggests a nucleic acid allows for distinct refolding and hinders of binding NB. To investigate if these transitions were related to the domains of denaturation or denaturation plus oligomerous association, we carried out a methodology of name SF in different concentrations. And here we can observe in the graphic of the tables of the melting one is stable independently of the type of the proteins in NA and NB. However, when we see the temperature for melting 2, we can see the raise in the concentration of the protein and the temperature in NA and NB in figure B. And to investigate if there are any effect of this protein, it was had a thermokinetic with different concentrations to see secondary studies, and we verified there was only one transition, and for both proteins NA and NB, independently of its concentration. And this melting temperature, it goes to 40 and 50 degrees, reinforcing the results that we had previously. And a paper that was published in 2018 showed that the protein had the capacity of making this dissociation and denaturize with the profile that we can observe in SARS-CoV-2. This way, we can suggest that temperature N1 is related to a process of denaturation and to the oligomers. We can conclude that the proteins and A and AB have the capacity of reverse this process, secondary and tertiarily as well. And we observed two transitions, and this was observed for the first time in this work, showing that this that was related to this naturation and dissociation of the oligomers, and these results confirm the leveling and the releveling of these proteins that are very common in viral proteins, and they are related to AN proteins and their capacities of having multi-functions. So we can investigate this phenomenon of dissociation of the oligomers, and it has several implications linked to a wide range potential of application of the protein. I would like to thank the laboratory Latea, Bio-Manguinhos, the partnership with Latea, Latin, that supplied with the proteins and antibodies, Bio-Manguinhos, the federal government, and Fiocruz.
Thank you, Luana. Congratulations on your presentation. Thank you very much. I would like now to invite all of you to pay a visit to our poster exhibit, and in the course of our event, vote on your favorite poster for the award, Popular Poster Award. You have to access the application of the event, clicking on the like icon, thumbs up, on the pages on the page of the poster that you have selected. The award ceremony will take place on the 10th in the afternoon. Now, continuing our presentations, I'd like to invite as the chair of the panel, Preparedness for the Future, Epidemics, Surveillance, and Scientific and Technological Development, the specialist of science, technology, and production and innovation and public health at Fiocruz, Julio Croda. He has a physician, graduated from the Federal University of Bahia. He did an infectology at São Paulo University, president of the ITB, the Brazilian Society of Tropical Medicine, and director of the Department of Immunizations and Communicable Diseases of the Ministry of Health. Currently, he's associate professor at the Medical School of the Federal University of Mato Grosso do Sul and the School of Public Health of the University of Yale. As the lecturers on this panel, we will have the director of the second department of the National Surveillance Agents, Meiruze Souza Freitas. Meiruze will make a presentation on the submission process, lessons learned from COVID-19. It will be done through a video. She is a pharmacist, graduated from the Federal University of Minas Gerais. She is a specialist in pharmaceutical technology. He worked at the Hospital Foundation of Minas Gerais and the Mayor's Office of Belo Horizonte and the scientific laboratories of the Santa Casa de Misericórdia Hospital. He is a civil servant working at Anvisa, National Surveillance Agency. And for presentations, I'd like to invite the senior official of the Vaccine Development Program on the Bill and Melinda Gates, Christopher Gill. He will make a presentation on the impacts of COVID-19 in Africa and lessons for the future. He is a physician and expert in infectious diseases from the Medical School of Massachusetts University and has a master's from the Tufts School of Graduate Biomedical Studies. He was a member of the teaching team of global health on the University of Boston, and clinical director for the conjugated vaccine against meningitis for Novartis Vaccines and Diagnostics. He got the Norman Scott Award for Excellence in Teaching at the Medical Public Health School at the University of Boston, and the Metcalf Award from the University of Boston, the highest award at the institution in teaching. We would also like to invite the scientific director of Biotecnopolo of Siena Foundation, Rino Pauli. He has a PhD in biological sciences from the University of Siena in Italy. He was a scientist and a guest scientist at the Rockefeller University at Harvard Medical School in the United States. He is the founder of the Institute of GISCAT Institute for Vaccines for Global Health. He is honorary professor at the Vaccines and Imperial College in London and molecular biology at the Siena University. He's a professor. He's one of the world scientific leaders dedicated to sustainability of global health. We will now give the floor to Julio Croda. Thank you. Good morning, everyone. Actually, it's a pleasure to be here with you.
Eu acho que assim, realmente esse
I
tópico
think that this topic is fundamental. If we will recall our recent past, in less than a year, we developed a vaccine specific to COVID-19, and that allowed us, in the first year of vaccination, it allowed us to, through this initiative, to save more than 20 million lives. And if we had achieved the goal of COVAT facility, we would have saved another 600,000 lives. So to have the capacity, response capacity to future pandemics is crucial. And this response capacity has to be more democratic, and its production mostly in the global south, where we have little infrastructure and capacity to respond to these pandemics. We, in Brazil, we had a very special situation, unique situation. We have two very large institutions that produce vaccines, both Bio-Manguinhos Fiocruz, as well as the Butantan Institute, which quickly allowed us to have vaccines, and they were able to offer those vaccines to the population of Brazil, and somehow they saved thousands of lives. The vaccine was the most important public health intervention in the response to the pandemic, and in future pandemics, it will remain the same. It's important for us to understand that the development, scientific and technological development associated with the vaccine is a matter of it's a security issue in a country from a point of view of getting ready, better prepared in the future. And we are living this future associated with climate change, the increase of diseases, the new diseases coming up, this closer relationship between the environment, human environment, and the spillover effects that we have noticed currently in so many situations. We have a classical example of this impact of climate change. We're going through the very worst outbreak of dengue fever, and an important response has come through vaccination. The Ministry of Health has adopted the dengue vaccine in Brazil, and at this very moment, unfortunately, the state of Rio Grande do Sul is going through the worst floods in history, and the impact that in a way will bring about the issue of infectious diseases and eventually issues of vaccine coverage and the communicable diseases through water and foodstuffs, leptospirosis, Bio-Manguinhos as a quick test for leptospirosis, and it's important for us to have this technological independence from the point of view of emergency response activities in the future. And this response must have an important local role, and in a way we must ensure access to the overall population, most importantly, the population of the Global South. I will bring to a close my initial remarks, and I'd like to invite our Meiruze to talk to us on video. She couldn't be here presently today, but she sent us a video, and I'd like the technicians to show us her video on the screen right now. Again, thank you, Meiruze, for your participation in this important symposium that's taking place in Rio de Janeiro. Thank you for everybody's presence, and we will start with the first lecturer on video. Good morning. I'd like to acknowledge the team at Fiocruz organizing this important event, this important debate. Increasingly, it's important to deal with and discuss preparations to face sanitary crises. We must not forget that Brazil and the world went through very many problems during the pandemic, and we have lessons learned to reduce the impact of the sanitary crisis, especially on the population, on the most vulnerable populations. We've learned a great deal through the COVID-19 pandemic. We exchanged so much knowledge and information and with officials around the world and NGOs and the World Health Organization. This exchange of information has favored the acceleration from the point of view of approval of COVID-19 vaccines and also the medicines that were used in the treatment of that COVID-19 pandemic. This is a time to forget the problems we've been through, but we must not forget that we're able to fight, to struggle against those problems, to overcome those problems. Science must come together from the point of view of regulation. I'd like to remind you it's so valuable to have the tools of continuous submission, continuous assessment of preclinical data, clinical data, technologies of having to do with vaccines and medications involved in the COVID-19 crisis. It's a learning that we must not overlook, and we must have it as a regulatory tool to prepare for new crises. I'd like to recall that recently Brazil has approved a regulation for pre-pandemic preparation vaccines, especially thinking of the crises linked to zoonotic influenza and the approval of a platform that may be updated if we are to face a crisis like the virus, influenza virus. That's the road ahead. We must think and discuss the approval of the platforms in order to allow us to do quicker updates as they're needed, be it in the presentation of a new pathogen or in the changes, especially the variations that occur with the new variants that have come up. How to update a vaccine, we must place it within our strategy, an update so that we may have data that will allow us to have approval, quick approval, effectiveness data, efficacy data, and security data, safety data. It's certainly a challenge to local officials to update these platforms on vaccines. And I'd like to recall the importance of effectiveness studies and real-life data used by Brazil and so many other countries when it comes to COVID vaccines, especially for the approval and the widening of age brackets. We must think of a design of real-life studies with robust data that are able to provide us with information for decision-making, regulatory decision-making, reliable data. So in addition to this continuous submission, we must look also at the guidelines on real-life data. We must remember that Brazil has established a real-life data guideline, and we discuss the preparation to face sanitary crises. And the context of data, real-life data also needs to have a scientific look together with a continuous submission data. We also have, we must make breakthroughs in the regulation and approval, perennial approval that will provide guidelines to face sanitary crisis when it comes to continuous data submission. We are at a juncture that we're discussing data for the presentation of the dengue vaccine developed by the Butantan Institute, which shows us that the regulatory tool is able to provide a celerity and a swiftness to the approval of the vaccines. And I'd like to remind you that in addition to the regulatory role to facilitate access, we must focus also upon a fair distribution, more equitable distribution that's able to reduce inequalities, especially in the access to such vaccines. That's also a learning that we must have from the COVID-19 pandemic. I'd like to, towards the end of my message, we must work together, but we must also be able to work towards a more equitable distribution, a fair distribution, and we must overcome these problems to the benefit of everyone, especially when it comes to sanitary crises. That's what I had to tell you this morning. Thank you so much for inviting Anvisa to talk today. And I'd like to salute Dr. Julio Croda, especially for putting together this wonderful event. I'd like to thank Meiruze for sending us this video. I'd like to excuse myself. She would like to excuse herself for her absence here, especially to present this topic and how the regulatory agencies in this process, mostly after the pandemic, how they worked from the point of view of speeding up the analyses that are crucial towards, at the end of the day, to provide quality vaccines to the whole population.
Presentation, I think you already have the question for the Christopher. Christopher talked about the impact of COVID in Africa and the lessons for the future. Thanks, Christopher, to come to Rio de Janeiro, and the floor is yours.
Olá, bom dia a todos. Unfortunately, that's all the Portuguese I know, but I'll try my best. First I wanted to thank the conference organizers for inviting me here. This is a wonderful event, and I feel a tremendous sense of honor here, especially to have the opportunity to share the stage with some of my old colleagues. Dr. Rappuoli and I worked for many years together at Novartis over a decade ago, so it's just great to be back here. Now, as you may have learned from my introduction, I'm now a senior program officer at the Bill and Melinda Gates Foundation in the Vaccine Development Group. And we're here to collaborate with Fiocruz and Bio-Manguinhos on the development of an MR and yellow fever vaccine. We're very, very excited about this project together. But actually, what I'm going to talk about today is some of the work that I did before I was on the granting side, but when I was a grantee from the Bill and Melinda Gates Foundation, working on some of our child survival and mortality surveillance projects in Zambia. And so I'm going to talk about the last pandemic as maybe some lessons learned for, I would say, the next pandemic, but I'm sort of hoping there won't be a next one, but it's possible there will be. So I start with this cartoon, and I suspect many of you have seen this in various varieties in the past. We start with a young gentleman crawling around on his hands and knees, looking underneath a light post, and the policeman comes along and says, you know, what are you doing here? And he says, I've dropped I guess he's saying, I can't say what he's dropped. It could be his keys, it could be money. And the policeman asked him, did he drop them here? And he says, no, I dropped them over there. And the policeman, of course, wants to know why. And he says, because this is where the light is. And we have seen this in countless variations, the so-called lamppost fallacy, where we look where the evidence is, but not necessarily using the evidence that we would like to have. Now, it's actually a very old analogy. I was curious about its origins when I was making this talk, and it turns out that the first use of this so-called lamppost fallacy goes back to the 13th century, to a fellow called Nasruddin Hoja, who was a Sufi mystic at the time. So, in case you were wondering, that's where it comes from. But I think I like this metaphor perhaps a little bit better, which is the traffic fallacy. And I think we're all very familiar with this, which is when you're driving at night and everything is dark and you're just looking at the highway and all you see is the cone of light from your own car. And how easy it is to imagine that that is all there is to see. When in fact, just outside of the beam of light, there's maybe a kid with a ball, or a dog, or a person on a horse, or someone on a bicycle, a car coming from sideways with its lights off. And so we fall into this safety sort of trap of thinking that we see the world when in fact we forget that we only see a tiny fraction of the world at any one time. And this leads us into this sort of logical fallacy where we take the absence of evidence for a thing as evidence of absence. We do this, unfortunately, all the time. So, and one of the examples was, of course, COVID-19. So I think I can use my little thing here. You'll notice that there's lots of red spots. This was from the Johns Hopkins COVID dashboard. You see all these red spots everywhere. These are where COVID cases were being reported at the time. This was at some point in the pandemic, doesn't really matter where. But the thing I really wanted to highlight was, where are the red spots in Africa? There really are none. And at the time, this led to some very interesting theories about why it was that COVID-19 skipped Africa. And I'll just recount a couple of the theories of this so-called Africa paradox. One was that there was a very high rate of cross-protection between COVID-19 and other circulating coronaviruses. It's an interesting theory. It's a little hard to reconcile given that these circulating coronaviruses are found everywhere else too. And so why wouldn't we also see cross-reactivity? So it's a strange theory to imagine that some coronaviruses only stay in Africa and other ones spread around the world. So and we now know that this is even though there is some evidence for this in terms of the laboratory, epidemiologically, this did not explain very much. Another one was that perhaps because of their experience with the Ebola responses, that maybe African disease control agencies were much more able to manage the COVID-19, but this is not true. There were the resources available to do this were simply inadequate. And in any case, how would you explain how the experience from Liberia and Sierra Leone and West Africa somehow magically populated the rest of the country? So I think this was not really true. The last theory, which I think is maybe quite an interesting one, is that the youthful population structure of Africa being much younger than the rest of the world perhaps allowed the populations to tolerate the disease better than elsewhere. And there may be some truth to this, in fact, and I think our data touch on this. Now, another theory, which was our theory, is that epidemiologic data about COVID in Africa were simply not there. And so we're simply looking at an example on a continental scale of the lighthouse fallacy, the lamppost fallacy. So the way we sort of stumbled into this investigation was a little bit unusual. We had been asked by the Gates Foundation to do a post-mortem surveillance study to look at the prevalence of respiratory syncytial virus in children who had died in Lusaka over a 3-year period. And the study involved essentially attempting to enroll every deceased child in the city during this period and then using PCR to test them to see if they had RSV or not. And so we had set up this infrastructure to do post-mortem surveillance for RSV. And then just as our RSV project was ending, the COVID pandemic began. And we went to our funders at the Gates Foundation and said, well, we have a post-mortem RSV surveillance study for infants. What if we turn that into a post-mortem COVID surveillance for all ages? And they said, good idea, because no one else was doing this at the time. So they gave us some money and we went ahead and did this. Now, in a sense, this was a very, very simple analysis. We weren't trying to model anything. We didn't use any really statistics except little n over big N, because we attempted to measure essentially all deaths in the city and to test them systematically. Now, what we ended up doing, just in case you're curious about the RSV study, since I mentioned it, I'll tell you what we found. The study eventually involved 2,286 deceased infants over 3 years, which was about 80% of the total in the surveillance period, and came from all parts of the city of Lusaka. And then we found RSV in about 7 or 9% of deceased infants. So from the Gates perspective, they were looking for evidence that RSV was having a major impact. This was quite clear evidence that RSV was having a major impact. So that's sort of where we ended on the RSV work. But now we had a skilled team on site. We had a molecular lab on site, which was staffed. We had protocols in place, including grief counseling protocols, which allowed us to engage very well with the families of the deceased. We had a team who were highly skilled in postmortem grief counseling. We had a pre-approved IRB protocol, which all we needed to do was to amend, to change the testing protocol, and then the age requirements, but everything else remained intact. And so we were basically able to convert the RSV study into a COVID study in about a month. And so we did. Now, what we found was that there were a great number of deaths, of course, during this period. This is a piece of data that allows us to sort of look at the representativeness of the sample that were enrolled in the COVID surveillance work, as opposed to all. And so what we see here is that in the blue are all deaths that occurred during this period. The red is the subsample that we tested from, and the gray were the individuals who tested COVID positive during this period. And so generally the three histograms are quite close, but you can see that there are some differences. Generally, if you break it down by age, you see that the prevalence of the COVID positivity rate amongst the deceased individuals tended to skew a little bit older than the total birth cohort, which is consistent with what we've seen elsewhere, that it was a disease that primarily struck the elderly. And so far these data looked very consistent with, in fact, what we'd seen practically everywhere else. We then sort of broke this down by age groups. We had, in the end of the study, this was just a subset that was done over a 3-month period, and so we had initially around 360, 370 deceased individuals. And out of that group of 370 individuals over that 3-month period, roughly 20% were testing positive for COVID. And so, as opposed to the then prevalent claims that there was no COVID causing mortality in Africa, we hear actually it was about 20%, which was very consistent with everywhere else. And we published this in the BMJ. I have to say, not that I want to blow my horn, but this was the most widely cited paper in my career. So it was a bit astonishing when this happened. We then went back to the Gates Foundation and asked them to extend the funding so we could continue to do surveillance, which they then did, which allowed us to look at 3 successive waves of COVID in Lusaka. So the first one was the one I initially described, which is here. I don't know if you can see that, but on the far left was that first 3 months of surveillance. And so you can see that there was an epidemic curve that we observed over time, then sort of tailed out. The red zone was between the 2 funding cycles where we had to stop surveillance. And so when the funding resumed in January, we were just catching the beginning of the beta epidemic, which was occurring in South Africa at the time. And this time, the COVID positivity rates up at the peak of the epidemic were approaching around 70% of all deceased bodies in the morgue in Lusaka. So, in fact, not just a common cause of death, but by far the most common cause of death that was occurring at the time. And then that second epidemic curve tailed off and resumed again in the spring of 2021 with the occurrence of Delta down in South Africa. And at this point, again, we saw this massive tail off. And that's where the initial study ended. Though I have to say we continued subsequently into the Omicron period, and I can talk about that if you want. But again, the prevalence of COVID in all 3 waves was extremely high. And when we did genetic sequencing, which is shown below in those little circles here, the first wave was the wild type, the second wave was almost exclusively beta, and the third wave was exclusively delta. The graph at the very beginning, at the bottom, the sort of multicolored images, was looking at a distribution of these cases adjusted for population in different parts of Lusaka. And the darker the colors represent where the disease was most concentrated. And so what I just want to highlight is, again, there's that little sort of empty area in the middle, which is the wealthiest part of the city. It's where the sort of the upper middle class of Lusaka live. Whereas the surrounding zones, much as in Rio, is where the poorest part of the population lives. And so what we're basically seeing, that just as it has elsewhere, this was a disease of poverty, even within a country that is very poor. There was still a gradient of poverty where it impacted the most vulnerable citizens in the country. And just to give you a sense of this is a satellite view of two of those compounds, the Chawama and the Kanyama compounds. If you were a histologist looking at this under a slide, it would look like liver. It is so densely packed with people. And this is where COVID hit hardest in Lusaka. All right, so a question that always came to us is, how do we know that these people died of COVID rather than died with COVID? So what we did to try to approach this was to assess the clinical symptoms that were present with each case. We assigned a probably due to COVID categorization if the person had presented with severe respiratory symptoms and fever and cough, respiratory distress, and a positive PCR. We said, that's probably COVID, a reasonable assumption. If they came in with a heart attack or a stroke that seemed anomalous, we said that might have been COVID because of sort of a secondary, thromboembolic event. And then the third category was probably not COVID, is when they had some clear other cause. They had, fallen down or had been hit by a bus or, died of cancer or something like that. Now, out of that first round of surveillance, we had 70 cases that were positive. Of those, we had 52 where there was enough data to assess the causality. Out of those 52, 44 out of the 52 had presented with fever, respiratory symptoms, or both. And so the majority were probably due to COVID. And only a minority were sort of explainable by other things. In the second round of surveillance after January, we now had 327 cases. Of those, 292 had enough data to make this sort of causality assessment. There were 73 pediatric cases in there, which we excluded because the presentation of COVID in the children was so completely different that we were really not confident that we could draw any sort of conclusions for that. And I think in hindsight, now that we know how COVID behaves so differently in children, that was probably the right assessment, which left us with 219 cases to adjudicate, and of those, 155 again, were possibly or probably due to COVID based on their presentation, so the vast majority. Now, it was much lower in the second round than in the first round, which was probably because there was some population immunity now built in after exposure to the wild type. So again, a very consistent effect. Now, we also were able to do minimally invasive tissue sampling on a subset. So we did transcutaneous lung biopsies using a spring-loaded biopsy needle. And in patients who had been assessed as having COVID before they died, which is a very small minority. This is a lung sample from one of those individuals who was diagnosed with COVID-19 pre-death. And this is a post-mortem lung sample, and you can see that this is a very diseased lung. The air spaces are filled with debris, and the walls are thickened. So this is quite consistent with a viral pneumonitis, i.e. COVID. So here's another patient with a similar pattern, and also some samples from the liver in two patients showing that it was not just a single disease, but this is a multisystemic event. Now, the question that then comes up is, why were so few cases being diagnosed by the government at the time? And so there's some answers to this. We start with, during that first round, we had 364 patients that were assessed and 70 were positive, so about 20%. Of those 70, only 7 had been diagnosed with COVID before they died. So about 10% had been diagnosed. But it wasn't that the other 90% were negative. The other 90% had not been tested at all. So we're looking at a tiny fraction who underwent any form of COVID testing before they died, even though they came into the ER with cough and respiratory symptoms in the middle of a pandemic. But they were not being tested. And the reason why is there were no tests. Full stop. They couldn't be tested because there was no PCR and there were no rapid tests at that time. So it was a complete absence of data. Even though if you spoke to the physicians there, they were like, yes, here's another patient who obviously has COVID, but none of those went into the official records for the government because there was no testing, a complete blind spot. Okay. Now, of the individuals who died in the community, which were the majority of individuals, none of them had been tested. So those 7 who were tested were all hospitalized mortalities. In the second round, we had 1,118 who were tested and 327 were positive. So about 30% on average were positive during the beta and the delta waves. Of those, 25 had been tested while they were still alive, which is still less than 10%, and all of those were in the hospital. And again, there was almost no testing being done in the community, even though 3/4 of the deaths that occurred were in the community. So another sort of structural bias in terms of what we saw. So why such a low rate of detection? The majority of deaths, a 4-to-1 ratio, were in the community where testing was not being performed at all. Among the hospitalized deaths, almost only half of those were ever tested. And unfortunately, in that second round where testing had become available, they were using rapid diagnostic tests, which are not very sensitive. So this was another sort of reason why. So overall, we estimated there was about a 90% underreporting of COVID deaths in Lusaka at this time. We then did some modeling based on burial registry data that we'd collected for 3 years before the pandemic and then during the pandemic, so we could look at an excess mortality analysis. And we did this with colleagues at the London School of Tropical Medicine. And I just want to draw your attention to that bump. That is the excess mortality bump associated with that first wave of the wild-type vaccine, of the wild-type virus. That is the beta, and that is the delta. And so in each case, there was a massive increase in all-cause mortality. This was separate from our testing, but was consistent with what you saw in Rio or in New York City or in Mumbai or many places which had the resources to look. Okay. And then this last figure shows that the majority of those deaths, again, occurred in the elderly. And when we looked at the cumulative effect, we were probably looking at around 2,000 excess deaths during that 1 year period from what would be expected on historical precedent. So, and the last thing I will mention is that during this period, because we were doing PCR testing and another group had been doing serologic testing, we had a positivity rate of the population. And we now had, through the burial registry, a total number of deaths rate. So we have N over N. And we could calculate a rough estimate of the infection fatality rate, which we did. And we found that the infection fatality rate for Lusaka was about the same as Belgium, which is to say it was average, not low, not paradoxical, not surprising, average. Okay, which is actually, when you think about, sort of profound, given what some of the very strident claims were being made at the time. It was just average. So, could Lusaka have been unique? Well, we only looked at one city. Could it have been unique? Yes, I suppose. We could have looked at the one place on the whole continent, other than South Africa, which was looking and finding where this was happening. But it would be strange that there was a magic force field around this one country and it was nowhere else. So we think that's unusual. We can also say that Lusaka had all of the ingredients for a pandemic: very crowded, very poor sanitation, very few, at the time there were almost no masks available. And so, you would imagine that it would spread very effectively then, and our data suggest it did. But this is true of Nairobi. This is true of almost any city in Africa. And we also knew from our own experience how expensive and time-consuming and difficult it had been for us to set up the surveillance system to study RSV 3 years before. And we had about a year to do that, with an expert panel helping us and lots of money from the Gates Foundation. Lusaka, the Ministry of Health in Zambia, did not have money and it did not have time to do any of these things. And so, of course, it couldn't. So chances are that, that lack of resources was not unique to Lusaka. So I would say it's possible that it was unique. And I only looked in one place, but I think it's unlikely. And I think therefore there are two hypotheses. One is that it was unique for some reason, and two is that it was everywhere. But that's where we looked. Okay, so I can't prove it. However, since that time, many other groups have done similar analyses and found actually the same thing. So this was the first opportunity to say, this Africa paradox thing sounds very fishy. Is that really true? And I think it was not true. And I'll end up here just saying, what would Sir William of Ockham have thought? This is William of Ockham. He was a British priest / philosopher. And logician, a fan of logic. And he is the one after whom Occam's razor is named. And if he were able to quote himself, he would have said, non sunt multiplicanda entias sede necessitate, which I don't speak Latin either, which basically means all other things being equal, the simple answer is usually correct. So my guess is that it was not unique to Lusaka. So what we do know is that COVID hit Lusaka very hard. At its peak, 90% of the people who were in the morgue were testing positive COVID-19. Most of the deaths that occurred were in the community where there was absolutely no surveillance at all. The emergence of the Beta and the Delta variants occurred almost exactly in parallel with what was happening in South Africa at the time. There was significant SS mortality concentrated in the elderly, as elsewhere, and the infection fatality rate was typical, just average. And so we think that the absence of reporting was due to a lack of surveillance and not some sort of magical effect that was unique to the continent of Africa. Now, I just mentioned in passing, great teams are built, they don't just happen. I want to mention two of our colleagues on this. One was Roy Chavuma, who died of COVID-19 in the middle of this. He was our site PI, who became sick on a Tuesday and was dead by Saturday. And was a good friend of mine. And then Magda Mwale, who was our project manager, who died of cervical cancer. And I will note that one of my main jobs now at the Gates Foundation is HPV vaccine research. So thank you very much. This is the rest of the team, and I appreciate your attention.
Thank you, Christopher, for nice presentation. And to point this important topic related to the surveillance. You need to have a strong surveillance to detect very quickly and to understand what is going on in the local settings, but also in the subnational level and national level. You have a time in the end for Q&A, but now I want to ask Rino to give the presentation related to new accelerated technology for diagnosis, vaccines, and treatment. Rino, the floor is yours, and in the end, you have time for questions.
Thank you. Well, it's great to be here with this full room of audience. And now we're going to switch gears. We're going to talk about technologies. And the technologies which were made available during the pandemic. And before I go to the technology, I wanted to share with you something which was very important during the pandemic that is not a technology on its own. And the slide I want to use to do that is this one. As many of you know, I've been spending 40 years in industry developing vaccines. And during my life, I always went to, basically, you start with discovery, then you do phase 1, then you do phase 2, then you do phase 3. And in all my vaccines, basically, takes between 10 and 20 years to develop a vaccine. And that's my experience. And then COVID comes, and basically, we couldn't wait 20 years. And the question was, how do we go fast? Obviously, we had technologies, but technologies were not enough. So one thing that happened was the governments, the public sector, came in and put money. This was true for the UK government, from Europe. But the most important one was the American one. The BARDA basically put down 12.5 billion out of the 15 billion that were invested by government. And then the guy who had the Moncef Slaoui, basically, who had was head of the BARDA of the development at that time, basically came to companies like us and said, well, you usually do vaccines by do discovery, and that takes time. Then when discovery is successful, you go to phase 1, and then when phase 1 is successful, you go to phase 2, and then go to phase 3. So, well, now, and basically the reason is you do that because you want to leverage your risk, financial risk. I mean, if you make all the investment up front, basically your company is going to go bankrupt. So he said, well, this time, use the check, use the money, the government is going to put the money into it. And then I'll tell you how you're going to do oops. It's not working anymore. Anyway, basically said, I'll tell you how we're going to do the development. You do discovery, And that may take 2 years. But as soon as you have the first thing that seems to work, you move to phase 1. And then phase 1 may take 18 months, 12, 20 months. But as soon as you have the first data, you move immediately to phase 2 and you start to check those of safety and so on. And then phase 2 sometimes can last 2 or 3 years. But you don't wait for the end. Immediately, as soon as you have an idea of the dose of the things, you move to phase 3, and that's what happened. And we had vaccines licensed in 10 months. Now, and that basically, what happened was that obviously the investment by the public sector accelerated enormously the development. Many people say, well, you went fast, so basically you took a risk. And indeed, a risk was taken, but was financial. The government took a financial risk because the things failed. But there was no risk on safety, efficacy, all these kind of things, because all the things were done in parallel, but not in and basically, in 2021, I was already using these slides. At that time, all the meetings were virtual. So we were with a panel of people, and somewhere in other place of the world, I don't know exactly where, there was a Nobel Prize in economy, Michael Kramer. I was saying, well, the big investment from the public sector basically accelerated vaccine development. And the guy, the economist, said, how long do you think it took to repay that investment of the government? And frankly, I was trying to scratch my head, I had no idea. And the guy says, 12 hours. I said, 12 hours? I said, yes. During that period, the pandemic was burning between 700 and 900 billion to the global economy per month. Accelerating vaccine development by 12 hours basically was a way to repay the investment. So that, I mean, nobody thinks too much about this. We always talk about technology, about things, but the economy of this is that. And then really that changed my mind, and then we started to work with another economist, David Bloom at Harvard, and we spent time asking, but is that true? I mean, can we actually put those numbers in the things? Because what happens is that if you follow this, the people that make the most money out of the pandemic, or out of vaccines, were not Moderna or Pfizer, but actually was the public sector. The public sector did the best investment ever made, financial investment. So we started to work on with David Bloom, and this is a paper which hopefully will come out soon, and basically started to calculate the internal rate of return for COVID, 561%. That's not bad, yeah, that's a good investment. And then we said, well, that was COVID, a special case, but what about a vaccine that nobody, all the commercial people all the companies have been, they say, group A strep will never be a commercial vaccine, will never make money. And so we started to calculate return of investment, 23%. Wow. And we started an impossible vaccine, like Alzheimer's. Impossible because it's very difficult, because we'll take hundreds of projects to make it, it will take many years. Well, even that will have a return on investment of 12%. So basically, this is just to say that it's not only technology, it's also the way we do things. And the fact that investing in health, in this case with vaccines, is actually one of the best things that government, the public sector can do. And is not only good because people are healthy, but because the return on investment is huge. Well, that was the past, and now what about the future? I mean, this is, you all know that, I mean, we just got off a pandemic. There is, basically, there are following climate change, the number of people on the planet, deforestation, urbanization, travel. We probably will need to be prepared for future pandemics, for future emerging diseases. This is a slide that's from Tony Fauci, it's not mine, but basically it tells you the complexity of the future, the things we need to prepare for. And I'm basically, right now I retired from the companies. I'm setting up a center for pandemic preparedness in Italy. And the question is, I mean, what do you do? I mean, there are so many bacteria, so many viruses, so many things. Which one you pick? I mean, and basically, since you cannot make vaccines or prepare everything for hundreds of viruses and many bacteria, there is one model which has been proposed by Barney Graham and now adopted by the basically by the NIH, and basically which is let's work on prototype vaccines. If you cannot make vaccines for hundreds of viruses because you don't know which one. So basically they divided the families of the virus in 30 different families, and then for each family they said, we'll pick one virus, which is typical of that family, and we'll go deep. We'll study that virus very well. We'll do the genomes. We'll make antibodies. We'll do the structure of the antigens. We'll basically design the antigen. We'll understand the correlates of protection. We'll talk with regulatory agencies from the beginning to the end so they understand exactly what they need to license the vaccine. And we'll make a prototype for one virus of that family. And we'll go so deep and understand everything that when another virus of the same family comes, we know everything and we move rapidly. So that's the approach that is being taken by most of the agencies, NIH, CEPI, and that's the one that we do. The one we do basically is the following. We start from people who are vaccinated or infected. We isolate monoclonal antibodies. And once you have monoclonal antibodies, you have immediately two things. You have something for prophylaxis and something for therapy. But at the same time, you know exactly what the monoclonal antibody recognizes. So now you can start designing a vaccine. And then you basically, with your monoclonal antibody, if you develop, you're going to figure out which are the clinical endpoints, which are the questions you need to answer to regulatory agencies for efficacy. And so basically, monoclonal antibody will allow immediate, basically, the approach or to the pathogen and accelerate vaccine development through the prototype approach. So that's the thing we do. And which are the technologies we're going to use? Well, the technologies I use, I think I put monoclonal antibodies there because they are a new thing that came out during the pandemic. During the pandemic, they did not last that much because they, unfortunately, basically, as soon as the monoclonal antibodies arrived, the new variants came and the antibody. But they are a new tool, very important. The thing is that they are fast. Here, for each of these technologies, I put two things, speed and equity, because I think they are two very important things. So I believe that antibodies are fast. I mean, the first monoclonal antibody was given to Trump in September 2020. That was before the vaccines, basically. You can do it very fast. And today, we can do faster. And I believe with the new technologies, in a few years, we'll be able to do much faster monoclonal antibodies, and we can drop the price and make them equitable, so affordable for high and low-income countries. RNA, well, RNA was fast, was the one that basically allowed rapid development, especially in the West, but was that equitable? Not at all. I mean, was only restricted to mostly to high-income countries. And what's going to happen in the future? When the future is going to be fast, it's still going to be equitable. I don't know. I don't know because still they're not stable, the RNA vaccines. They're more stable than they used to be, but they're not completely stable. Still the cost is very high. So hopefully, technologies usually with time, they become better, cheaper and things. But I'm not sure in a few years whether RNA will still be an equitable vaccine. Classical vaccines made by subunit adjuvants. Well, in this pandemic, more or less, they've been a disaster because they arrived when the pandemic was over. So very disappointing. So they were very slow. I mean, they've been shown to be efficacious, they've been safer, less reactogenic, stable at the refrigerator temperature. So they're very good, but in this pandemic, they were not really useful. What's going to happen in a few years? I think the technology, I mean, the fact that RNA vaccines were so fast basically put the pressure on being faster with protein-based vaccines. So I expect in the next few years, we'll find ways to basically to accelerate the development of protein-based vaccines with adjuvants. And I expect that if there is a new emerging thing in the next few years, protein-based vaccines will be there running at the same time with RNA. And then we have viral vectors. Well, viral vectors were extremely fast, as fast as RNA. They were equitable. This is the only ones that really made, or the majority of the non-Western world population has been immunized with viral vectors, so extremely important. They did not make too much in United States and other places for regulatory and maybe safety reasons. In the future, will they be fast? Yes, they will continue to be fast. Will they be used equitable? Here, I think the equity is going to be inverted because they're not very unlikely will be available in places like United States, other places, because of regulatory things. But they will be for sure available in other places. So those are my considerations for the technology of the future. Technologies are there. I mean, they will be available. They can go fast. They can make equitable vaccines. In addition to vaccines and technology, we have monoclonal antibodies. I think that will be part of the future in dealing with infectious diseases. But in addition to technology, remember the economy I mentioned at the beginning is extremely important. And with that, I think I want to thank you for your attention.
Thank you, Rino, for the presentation. You have time for Q&A. Se alguém tiver alguma questão, fique à vontade. Eu vou começar.
Feel free to ask them. I'm going to start off with a question.
Christopher, to give some update about the Omicron wave in Lusaka, how the thing is going on related to the deaths, especially related to the deaths, and how the vaccine covered nowadays in Lusaka, especially for vulnerable population.
Thank you for that question. Obviously, during the first wave, which was 2020, there were no vaccines anywhere. So equity was not an issue. But in the United States, we started to have access to the AstraZeneca vaccine around January or February, I think it was, of 2021. Our colleague Roy died during that window. And I think the first member of our team to get vaccinated was in the fall of 2021, so almost a year after they first became available. And he had special access to the AstraZeneca vaccine. He may even have received it out of Zambia, I'm not sure. Widespread availability of the vaccine has never really happened. And post-pandemic, of course, as elsewhere, we see this paradoxical rejection of COVID vaccines where very few companies can sell them or almost give them away at this point. So we've gone from tremendous demand to no demand and almost complacency, which is an interesting lesson learned and makes me worry about what happens next time again. So thank you.
Thank you. I have one question for Rino also. It's also related to the first question. You point very nice that probably the monoclonal will be alternative in the future if it become more cheaper, for sure. But nowadays you have this discussion, especially related to RSV. You have two options nowadays, the monoclonal and the vaccine for RSV. And you have a difficult for supply, especially in the North Global. The countries try to buy this monoclonal and then they don't have access. And how you can deal with this for the future, because I think you still have some problem in the scale up this production, and you have a good example nowadays. Probably for us, in the end, you have access, especially in the private sector, only for vaccine in the pregnant human, and don't have access to the monoclonal for the children that provide a good protection also. I want to you that you can comment a little bit about this, how scale up the monoclonal production and use RSV as example for this. That's occurred in this year, and you can discuss this.
It's an excellent question. In my slide, I said monoclonal antibodies today are not equitable. I mean, there's no way you can, because they're producing mammalian cells, the production is very high. All the development of monoclonal antibodies is done for cancer, where you have high price, low volumes, and what we want is high volumes, low prices. And the technology, as it stands today, is not there. Now, we know that since a while. A few years ago, I pushed the Wellcome Trust to come up with a booklet saying we need to make monoclonal antibodies equitable. Now, to do that, you need to go beyond the technologies that are there today. And there are technology where the alternative expression systems that could actually do that. So that's one of the goals, one of the things I want to do in my Center for Pandemic Preparedness. Basically, develop the technologies that will make these very important tools available and accessible in low-income countries as well.
Thank you. Temos uma pergunta.
Luiz Castelo Branco, Fundação Ataúfo de Paiva. My question is for you. Taking in concerning that COVID-19 is a mucosal disease, I would like to know what you think about vaccines and different routes of immunization, taking in consideration that maybe you avoid infection as well. That's one of the most important things in relation to COVID-19. So that's my question.
Maybe Rino can...
Yeah, no, you have an excellent point. I mean, what we learned is that let's put it this way, if the virus had not changed with the variants, probably the systemic immunization was going to provide lifelong immunity. But since the virus changes, basically you keep a protection from systemic disease, but basically every few months after the last immunization, you become susceptible again to infection. So the big question is, do we need different vaccines? And obviously it would be nice to have different vaccines, especially mucosal vaccines, especially intranasal, oral, are delivered by aerosol, delivered by, and so that's what people are trying to do right now. There are initiatives in China, in India, and some in the Western world, where they're trying to make vaccines mostly delivered by viral vectors, some of the adenovirus, but also other viruses being tested, that could induce local immunity. If you induce local B and T cells, mucosal sites, probably you can protect from infection. Is where the development of new vaccines is focusing right now. We'll need to wait some time to see the results of these studies.
I think you finished this section, the first section for your meeting, and I'd like to thank you both and Meiruze that speak by video. And I think you have an interval now for the post presentation, and you return in the next section. Thank you. Thank you again.
Sirs, thank you so much for the presentation. Obrigada, Julio Croda, pela sua moderação.
Thank you, Julio Croda, for your moderation. Before going to the interval, I would like to make a quick remark. We're going to have a 25 minutes interval, and I would like to ask you that punctually return and do not refrain from doing the poster visitation, the exhibition, and also the booth that is in the external area. Please, in 25 minutes. Thank you very much.
Próximo painel, o diretor de Bio-Manguinhos,
The
senhor.
next panel, the director of Bio-Manguinhos, Mr. Mauricio Zuma. He has a degree in administration. He's a doctor in innovation technology in Sussex University in England and master in management of technology innovation in Fiocruz. He has an MBA by the Federal University of Rio de Janeiro and specialization in public administration in Brazilian Administration School of the Getúlio Vargas Foundation. I pass the floor to Mr. Mauricio Zuma.
Alô? Olá a todos.
Hello, everyone. So I ask you to wait just a moment to announce. We have announced for everyone who is entering the room yet. Just a second, please.
Então vamos lá, pessoal. Bom,
So we can start now. So this panel, we're going to do it in English, okay? So those who need the device for translation, please get it with the team.
hello everyone. Thanks a lot for coming over. It's a pleasure to be here and to see so many people participating in this event. It's amazing to see so many people interested in our events. So thanks a lot. And before entering the panel itself, I want to express my solidarity to the people of Rio Grande do Sul for the flood that has devastated several parts of the state. So it's very sad thing. So my solidarity, our solidarity for them. And just to let you know, it will be announced that I believe it will be announced here. There will be some collecting points for donations during the event outside. So if anyone wants to donate anything, there will be points for collecting the donations. Well, starting about our panel it's about DCVMN, presented by the members of its board. So for those who doesn't know what DCVMN is, DCVMN stands for Developing Countries Vaccine Manufacturers Network, a network founded almost 25 years ago, and with the objective to promote the development of the vaccine manufacturers of the Global South, and to allow their participation in the debates at the WHO. Dr. Akira here is one of the founders, And over these years, there has been a lot of development and growth of these manufacturers, and culminating with an important strategic role during the pandemic. As well as now important manufacturers, vaccine manufacturers, we are also important innovators. So I'm not going to anticipate what will be said during the presentation, but this is just a brief introduction about the DCVMN. Well, let me tell you that I'm very happy that we will have here all the board of the DCVMN participating in this panel. I'm very glad to share with all of them this stage, and it shows the engagement and the commitment of all of us in the success of our network. Okay, that said, let me call all the participants of our panel here, starting by Dr. Adriansjah. Please, Dr. Adriansjah. Azhari. Dr. Adriansjah is the chair of our board and Senior Executive Vice President of the Biofarma Indonesian manufacturer. Thank you, Dr. Adriansjah. Let me call here Dr. Tiago Rocca, Vice Chair of our board and Business Director of our brother institution, Butantan Institute. And let me call also Dr. Rachel Park, Director of International Business at Gill Biologicals, a manufacturer of South Korea. South Korea is no longer a developing country, but it's a pleasure to count on them, still on them in our network. Let me call also Dr. Venkatraman, Vice President of International Business at Bharat Biotech, important Indian manufacturer. Let me also call here Dr. Andrew Wong, CEO of the Chinese manufacturer Walvax. Also, Dr. Fernando Lobos, Director and Deputy CEO of Synergion Biotech, Argentinian manufacturer. And I think it's virtually. Is there anyone? Virtually? Yes, oh, thank you. Dr. Sunil Gairola. Dr. Sunil Gairola is Executive Director of the Serum Institute of India, simply nowadays the biggest manufacturer of vaccine manufacturing in the world. And at last, not least, Mr. Rajinder Suri, please, the CEO of our network, the DCVMN. Well, our panel will start with a presentation by Mr. Rajinder Suri, and then after that, I will make at least one question for each of the participants of our panel. And if you have time, I will open for questions from the audience. Mr. Suri, the floor is yours.
Okay, so thank you very much, Professor Mauricio Zuma, for this kind invitation, and on behalf of the entire board of DCVMN, I take this opportunity to thank him profusely for inviting us to join this illustrious audience of intellectuals to discuss what could be probably the most important aspects of human health, the vaccines. And we saw this morning, we heard the best of the speakers, and I'm really very, very honored to be part of this, high-value forum. So today, when we are talking about DCVMN, Mauricio actually stole some of the words that I wanted to use. So excuse me if I repeat that, but I would definitely like to introduce DCVMN to you first of all, so that we are at the same page, same wavelength, and we know what we are talking about. As far as DCVMN is concerned, the Developing Country Vaccine Manufacturers Network, I see my friend Solo in the audience, so it's a pleasure to have him here. So as far as DCVMN is concerned, let me have, I don't know how to move the slide. Let me see if I can do that. Yes, it moves. So we are a network of 45 vaccine manufacturers engaged in vaccine innovation, R&D, production, and finally delivery. We represent 16 countries across all 6 WHO regions, and I am very happy to share with you that we have been supplying to over 170 countries worldwide. Pre-COVID, the total capacity of developing country vaccine manufacturers was resting at 3.5 billion doses, which is today at 14 billion doses, 4 times. I think this deserves a big clap from all of you. In fact, I am very pleased to share again that we deployed all the platforms that could be used for COVID-19 during the pandemic, whether it was adenovirus-based non-replicating or replicating viral vector vaccines or nucleic acid, both DNA and RNA vaccines. In fact, we were the first to develop worldwide a DNA vaccine which was used for COVID-19. And of course, we have used very frequently the live attenuated vaccines as also the protein subunit vaccines. In fact, the best thing that happened during COVID amongst its devastating impact was that there was a collaboration worldwide in terms of international technology transfers with developing country vaccine manufacturers. To quote some of them, for example, AstraZeneca with the Serum Institute of India and SK Bio in Korea, and J&J USA with BioE in India and Aspen in South Africa, whereas Pfizer had a tie-up with BioVac in South Africa. Now, coming closer home, we are very happy to announce that there were several South-South collaborations within the developing country vaccine manufacturers, and these were, between Sinovac to Vaxira to PT Biopharma to, and Sinopharma, for example, had with Sodema and PT Chemia, and Serum Institute of India had a collaboration with Aspen in South Africa and Vaxira in Egypt. And within India, Bharat Biotech had a drug substance manufacturing collaboration with Indian Immunologicals. I think this is the best success story that we can think of and write home about. It was a global ramp-up of vaccine production by the industry from 0 dose in December 2020 to within 1 year, they ramped up to 11 billion doses in less than 324 days, and then taking it to 13.59 billion doses, out of which I think you will be happy to note that more than 60% contribution came from the developing country vaccine manufacturers. So there were very clearly few enablers of the ecosystem, whether it is technology, whether it is regulatory harmonization, or establishing the framework. In terms of technology, I must say that it was early access to technology, IP and know-how, because IP alone could not have helped anybody. Even if I hand over to you on a pen drive the IP of a product, you will not be able to make vaccines. So what is important is the hand-holding, and that is what happened during the technology transfers. Then, of course, is the regulatory harmonization, and here I think we all have to thank our regulators throughout, whether it is US FDA, whether it is EMA, or WHO, or Anvisa, and several regulatory agencies within the countries who really helped to expedite. They were working round the clock. On 31st of December 2020, when all of us were celebrating New Year Eve, the regulators were sitting in their offices and giving approval to the vaccines. So thanks to them that we had the vaccines on time and much earlier than anticipated. Should move. I don't know why it is not moving. Sorry. Yes. So this is, the success story chapter which I wanted to share with you, that out of 13.59 billion doses, 8 billion doses have been contributed by the developing country vaccine manufacturers. I think this needs a little bit of a big hand from all of you. So this is a clear, demonstration of the fact that developing countries could contribute much more than was anticipated. I was speaking in US few months back, and it was surprising to know that many people did not know any other company other than Pfizer and Moderna. And I think this is something which all of us have a challenge to prove to the world, that what developing country vaccine manufacturers can contribute and can stand for. I think the cells of this have gone down. No. I don't know. Can somebody help? Just stay here. Yeah. Yes. Okay, done. Thank you. So, some of the challenges that we encountered, of course, all of you are aware about the shortages of raw materials and packaging materials right in the beginning of the COVID-19 that, created havoc, because you must appreciate that each vaccine requires more than 100 to 200 components to manufacture and get delivered in the marketplace. And even if there is shortage of one particular item, the whole value chain comes to a grinding halt. And this was something which was amazing, that at that time the DPA, the Defense Production Act, was put in by US, and the materials, particularly the single-use materials which were being manufactured only in US and some countries in Europe, were totally restricted to the other countries. The second, of course, was sustained investments and funding, and probably you will not have this information, that many companies, including Moderna, did not have access to funds very early in the stage, and it was risk funding which was probably came to the rescue of everybody, because the manufacturers started investing. For example, Serum Institute of India had built up an inventory of first 10 million doses and raising it to 50 million doses. Even if you consider it is $3 a dose, it is $150 million investment at risk before it gets approved. So I think this requires a lot of guts and a lot of vision And thanks to Mr. Adar Punawala that we had this vaccine very early in the game. And then, of course, the virus was playing tricks with, continuously moving from one variant to other, and there were several mutations. One of the biggest challenges that I think developing country vaccine manufacturers faced, Raleigh in the morning talked about $15 investment, $15 billion investment in COVID-19 vaccines. Out of that, $5.6 billion was invested in 1 year, first year 2021. And out of that, just 5% came to the developing country vaccine manufacturers. And this is something all of us should be aware that we contributed 60% of the total volumes and we got only 5% of the allocation of the funds. And I think this is something which we need to really stand for and go and raise our voice share. And that's what is really the role of developing country vaccine manufacturers today. That's what we are doing. Because there is a need for reflection for this disproportionate allocation versus the success rate of the deployed funding. So there were several lessons which we learned during the COVID-19 pandemic. And of course, the goal stays to develop a holistic approach, including but not limited to, the positive predictive models and a much faster, access to strains, and of course the funding, access to funding, which was, as I said, was more of risk funding, and definitely sustainable business partnerships, which is going to play a big role. Talking of pandemic accord, which is, I think, going to be one of the major milestones going forward for any public health emergency of international concern, it is important to note that, INB has been pursuing this. Of course, there have been some bottlenecks which have been encountered, but negotiations are going on, and we are very hopeful that something very positive will come out of a pandemic accord, which will make sure that you have sustainable supply of man material and machines when, without any restrictions or boundaries. And then, of course, speed and scale are important milestones, and CEPI's model of 100 days mission is going to be one of a landmark cornerstone in this direction. I must talk about investment in capacity building, and capacity building is not only in manufacturing facilities, but also in terms of building the manpower. And I think that is what is very important, and DCVMN is playing a big role in that. I'll just come to that in a few minutes. So what are the cardinal principles for successful technology transfer? Of course, it has to be the culture of the organizations involved, and I use an acronym LMP, which is like-minded people, because even if you have the best of technologies, they will not work unless you are mentally connected. And a mutual trust and transparency, because we have seen that the very, very important technology transfers have also failed because there was no trust between the partners. A rock-solid contract which envisions all the challenges and solutions are built into that, and this requires a lot of experience and expertise. And of course, we need to communicate with each other, we need to communicate with partners, we need to communicate with our managements, with the boards, with the employees, so everybody has to be on one page. And the most important, again, is the transfer of technical know-how, which I said is end-to-end hand-holding, which is critical for any tech transfer. So we at DCVMN, stand committed to innovation, to capacity building, in manufacturing in LATAM, in Africa, and also in other regions. And I was just speaking, that we will be working hard to make sure that there is an access to TB vaccine tech transfer at the earliest possible. And in terms of collaborations with stakeholders such as WHO, Gavi, UNICEF, PAHO, and various other organizations like BMGF, CEPI, PATH, and several others. So I stand at your disposal to make sure that your voice is heard, and we have made sure in the last 3 years this voice is reaching the right lobby, and things are happening. They have started begin to happen, and I seek your good wishes for the future. Thank you very much for your attention.
Thank you very much, Mr. Suri. It's very impressive, the presentation. I listened to this presentation several times before, and I always get proud of the achievements of our network. And now, as you are all aware about the key role of our network, I will start making some questions to our guests. Starting by Dr. Adriansjah. Dr. Adriansjah, what are the elements in strengthening the global vaccine ecosystem?
Thank you very much for the question. Firstly, I'd like to thank you for inviting DCVMN for this very prestigious meeting. So yes, based on lessons learned from the pandemic crisis, we see that more attention has now focused on how can we strengthen the ecosystem of vaccine in general. You see now, in terms of ecosystem, there are two types. One is internal, regarding the vaccine industry itself, and one external, involving other stakeholders here. So from the internal part of the vaccine industry, you see that the first and very most important role is R&D, because R&D here is one part that needs to be well exploited in terms of tech transfer, and also how can we again access to technology in the fastest and easiest way. And one of the challenging items in terms of tech transfer is of course IP, and hopefully this can be done in relation to the crisis of this pandemic. And secondly, what we need to do is that having gone through R&D, we have to also have very, very well and defined clinical trials, because clinical trial development is very important to know the safety and efficacy of one vaccine. So we know that many trials have been done, basically done in LMIC countries. So countries in the developing world has the very good potential to execute clinical trial sites when need be. So I think it's a very good alignment for us to be in line with North and South collaboration. And once we have that, we go into now manufacturing, where we have a lot of investments done need to increase capacity building, because you know that the need for vaccine deployment is very high. So manufacturing is one part that needs to be done in great care, because of course investment has to be done, and the platform that we're going to use is also one point that asks for us to engage in the technology that we're going to invest in terms of facility. Now, of course, when you go into distribution, one of the most key elements in vaccine is the cold chain system. We see that based on lessons learned from the pandemic, we have a variety of temperature from vaccines from 2 to 8 until -70. So this can be a big challenge for us, for public sector entity, to try to have this vaccine deployed in areas. For example, in Indonesia, we have 13,000 islands, and we have to distribute this vaccine equally among islands. And this is one part that a cold chain distribution system has to be well in place. And of course, in terms of the traceability of this vaccine, a good track and trace system has to be embedded, because we can know where does the vaccine come from and where does it go. And this is also one point for us to be able to be engaged in terms of the ecosystem in the industry itself. Outside of that industry, of course, the regulatory harmonization, regulatory expeditment is one important part, and also the policy makers that can exploit this vaccine is one important factor for us to be engaged. Of course, and also the vendors that are supporting the raw materials has to be engaged in this whole ecosystem. Thank you very much.
Thank you, Dr. Adriansjah. Tiago, in your opinion, what do we need to strengthen our capabilities, innovation capabilities here in Brazil, in order to strengthen our role to not only to the Brazilian public health, but also for the regional public health?
Thank you, Mauricio, and thanks for bringing this opportunity for the DCVMN board to be here in Brazil. We are very happy and very proud for this moment. I think the unique in the last 5 years that I'm in this journey. But now I'd like to excuse the board to answer the question in Portuguese to my attendance here, okay? I don't know if you want to connect here, but I'd like to reply this in Portuguese. Bom dia, pessoal. É um prazer representar não só o Butantan, mas o Brasil dentro desse conselho de membros, né, da indústria, de fabricação.
Of the industry of manufacturers of vaccines. And with respect to Mauricio's question, I think it's important for us to leave a message here. You saw in the presentations by Mr. Suri and some figures that he alluded to. What do we must notice in Brazil that we're lagging behind the other countries and the manufacturers on this block of countries, especially South Korea, India, China. So we have a long way ahead in terms of growth, not only in innovation, but also in capacity building and industrial capacity. And we have the challenge of catering to the needs of our public health system. That's the greatest legacy we have, the greatest volume of vaccines and demand with new vaccines coming in. That's the opportunity we have to reverse that trend with a great deal of investments, mostly in innovation. We have many gaps to bridge, innovation that we must fill those gaps, technological platforms that we don't have access to, services that we depend on the high external hiring to provide the needs for laboratories, and we end up getting in line, and that's what holds projects back, the lack of resources and also the lack of time, and that leads innovation projects to the Death Valley, if you will. It takes them to the lowest depths of the Death Valley. Bridging those gaps requires investment so the projects won't die on the way to in the course of their trajectory. We need to ensure investments that won't, it has to be a state policy. It has to be embraced by the current government, but it has to be built into a state policy so we won't have to revisit this issue in 4 years' time. China and India and South Korea, they started investing for a long time and provide building a human capital. There is a work on ensuring that Brazilian scientists will be repatriated. For example, there was a lot of brain drain. They went to work abroad, and we must bring them back. And that's exactly what India, China, and South Korea have been doing. The dengue epidemics that we're currently going through will be a leverage to increase our perception as a Brazilian representative on this council. I see that on this board, I see that that's what we have to do to get to the same level that these companies in other countries have reached. So thank you.
Rachel, as I said before, the South Korea is no longer a developed country. It's a high-income country now. Developing, it's now a high-income country, but you are still making part of our board. My question is, our network. My question is, in your perspective, how South Korean regional manufacturers acted in response to the pandemic for vaccine security? And how about the lessons learned?
So thank you for your question. Also, thank you for extending invitation to all the board of directors and members of DCVMN to this very important gathering. So though South Korea was the third country which is in-house developed the vaccine and therapeutics licensed for the pandemic in the world, and I must acknowledge that Korea was the latecomer to the market as the first vaccine was only licensed in July 2022. However, as Dr. Rajinder Suri stated in his presentation, the collaboration between Korean manufacturers and multinationals was very active for the production of vaccines and therapeutics for equitable access during the pandemic. So as you may have heard, as Suri presented, a few notable collaboration with the big players of South Korea was Samsung Biologics with Moderna and SK Bioscience with AstraZeneca and Novavax. So South Koreans were able to have access to locally manufactured vaccines through the collaborating with multinationals. And also we pursued the dual strategies in parallel, which means that we produce the vaccines in collaboration with the multinationals while developing our own vaccines. So as you may have heard, then Korea experienced Korean War in 1950, and nothing was left. And with the ashes, we developed. There's an economic development led by a strong leader, and then we become one of the OECD countries now. So we do have a 빨리빨리 nature. 빨리빨리 means immediately, as soon as possible culture. So which was one of the drivers for fast economic development of South Korea. And same thing can be applied to manufacturing business of South Korea, including the biopharmaceuticals. So in addition to this, and South Korean manufacturers have a facility and capabilities in compliance with the CGMP standards at relatively lower cost. And also South Korea has a very stringent national regulatory system, so Ministry of Food and Drug Safety, which was listed as one of the first 3 WHO listed authorities recently. And for vaccine development and funding support was available not only from internal, but also from external sources, global stakeholders, and South Korean government. So they enable us to act promptly and actively during the pandemic. So in that sense, and I think strengthening regional production capabilities in response to emerging demands, as the session title says, is critical. So enhancing the vaccine development and technology transfer, the partnership is key. So partnership between manufacturers and developers and with different stakeholders, for example, public, private funders, and regional, supranational regulators and policymakers. And in particular, I have to echo what Suri presented. And sustainable funding is a key for developing countries' vaccine manufacturers network as a tool of risk sharing. Thank you.
Thank you very much, Rachel. Now let's make a question to Sunil, Dr. Sunil Gairola. Dr. Sunil, are you still there? Oh, yeah. You are not hearing you.
Am I audible?
Okay. Now okay. Dr. Sunil, Serum Institute is a partner to Regional Production Vaccine Initiative for Global South vaccine demands. Please provide us an overview of Serum Institute partnerships for such initiatives.
Thanks for, Mauricio, thanks for your question. And really, I feel so bad that my all colleagues are there as a part of board, and I couldn't attend it. And Rio is a place where I would have definitely loved to come. But anyway, maybe next time, whenever there is a meeting happening in Sao Paulo as a AGM, I'll be there. And I'll definitely try to see Rio. Your question is with respect to serum philosophy. I consider, if you think on one line, that serum has always worked, it has a different business model. And it has a philosophical in a way that serum always wanted a product to be made accessible to all the people who really need them the most. And that the moment serum has come in any manufacturing, they've always worked on a large scale. And that scale has gone to such an extent that, of course, the vaccine doses have been made available to the maximum people, which has happened in case of COVID also. And if you see the conventional vaccine, where I really appreciate Mr. Suri for giving such a nice presentation and giving us a boost also, I would say, as a board member, that we are doing so much. It is really surprising also, sometimes it happens. And we are only the 5% benefiter of all the, donations or the funding which is given, which is really very challenging and very alarming also. Where we can supply, we are supplying more than 60% of the vaccine doses. So I really appreciate Mr. Suri, our CEO, who has really taken this task, and he is doing a very sort of a I would say nice job, but he is doing for humanity that whosoever is the one who can really supply a material, who have a potential to supply a material, why they should be deprived of those benefits which others are getting. So this is my first take on it. And if you see serum's contribution, I would say that you can take any product which is there. Serum has always done the maximum availability of the product, and they have reached from one place to another in Africa or in Europe, but they have supplied all the product and they have kept their promise. The stockpiling capacity, the challenge to have what you called a risk, which Mr. Suri has already praised our CEO, Mr. Adar Poonawala, that he has taken that risk of just having a product. The moment it is approved or it is getting a what you call regulatory nod, we are able to supply. So that risk is not because of the money, that risk is because of the humanity. Because we wanted to serve humanity, that's why we have done it. And I'm very lucky to have working with, as a board also, with the Serum Institute, because we have those philosophies in place. And secondly, if you see that Serum has always made a product and they have always gone for a pre-qualification. And pre-qualification is the one which has a wider reach in terms of supplying a product. And best example is the Manafrivac. You can imagine that that was only for the sub-Saharan African countries. And maximum challenge was that the vaccine has to be supplied at 40 cents. So you can imagine it's a lifelike product with a diluent still at 40 cents. So it was really a call on only humanity. So Serum has worked on it and Serum has delivered that product. Now more than 300 million doses have been given. And you can imagine that now MenA disease is completely gone out of that. But unfortunately, the bacteria has its own way of living a life. So now there is a C is coming up or there is a Axis is coming up. So then again, we have taken a call that we will go for a polymanage. Now polymanage is a pentavalent. So all products which needs to be as a part of making the humanity and human health well in terms of meningitis cases, we have developed that product. And today that product is a WHO pre-qualified product. So you can, one thing you can definitely see that there need to be a partnership which is happening. And for MenA, there were at least, I would say, a path was there, WHO was there, foundation was there, and it was a very great partnership. So unless we believe on partnership, unless we believe on scientific sharing, unless we have a belief on ourselves, I think this cannot be done, but this is being done by the DCVMN members. And I'm very sure that even newer products which are coming up, we have a very good role to play. And as far as my say on the partnership which is happening with the African countries or Global South, I would say that we are already in partnership with Aspen, we are already in partnership with the DAK, and Vaxira, of course, as Mr. Suri told about it, Senegal. So these are different products which are happening. And one thing I must appreciate on the part of the African regulators. Normally, what happens that once you have a product approval from a regulation which is little higher, which we say as a EMA or USFDA, then there is an approval from the other side. But R21 malaria case is a true example how the sovereignty in terms of the regulation has gone up. We got an approval from NAFDAC. We got an approval from Ghana. Ghana was the first one to give. So you can imagine their evaluation and the need, they have done it. And this is a very good sign in terms of what I say that the regulatory framework, which is getting improved, will definitely give a sort of a surpass and very good sort of a advantage to all the manufacturers who are there. So those are the things which are happening, and we are very happy about it. Of course, now R21, we have got a WHO PQ. And as I told you that we always want to go for WHO PQ because we want to reach to the maximum and reach to the real needy who wants a product. Now Ghana and Nigeria, they have approved our product even before WHO PQ, really very strong. And the vaccine is now approved from 5 months to 3 years of age. So malaria is one story. And can you imagine the moment serum enters into the field? Of course, RTS is available as a product, but the only gap which is happening is the accessibility, the availability of the product. And you have to definitely think about if it is 10 million or 20 million, what is the population need to be immunized, and what is the population which still will have a gap in it. But when I say that serum can give 150 to 200 million doses. So everyone in the countries in Africa or in the countries where the product is needed, they can definitely plan their immunization schedule to such an extent that each and individual is protected. So this is where serum has a very big contribution in that. And I will definitely say that serum is always in support of the local production, which happens in the African continent. It's all now in public. We have a good coordination with one of the Ghana companies called DEK, and Aspin, is already known. But then if you see the Ghana and Aspin, it's a total different philosophy. Aspin is already something which is established. They've already made 1.5 million doses of J&J product. So they have that knowledge of doing it. But the moment we come to DEK, we have started a collaboration from a groundbreaking. So we are involved with them from designing a facility and of course to the erection of a facility, and it's a long-term project. So serum has never restricted in terms of that this technology is only for serum. No, we wanted to share the technology. We want to work for humanity, and this is being done. So I think in short, I would say that these are the two diversities. Aspin, little advanced in terms of J&J product they have made, and they have the facilities, and DEK just starting as a groundbreaking things. So that way we are sharing our knowledge, sharing our technology, and we would love to have that each individual who is needy, requires a product, should get a product. And that's the philosophy I think serum has, which is, I won't say only philanthropy, but yes, it is something which is very much humanitarian. And I will wait for the next questions if are there. That's where I close my answer to this. Thank you.
Thank you, Dr. Sunil. The capacity of Serum Institute is always impressive. Yeah. Let me turn now to this side, although we are all at the same side. Yeah. I will start by Dr. Venkatraman from Bharat Institute. Dr. Venkat, how can vaccine manufacturers collaborate in the light of regional vaccine manufacturing programs to benefit the entire global south while addressing vaccine inequities?
Good afternoon, ladies and gentlemen. Thank you, Dr. Zuma. Interesting question. Let me answer this in two parts. We are talking of inequity, inequity, and we are talking of collaboration. I think it's important that we understand this term inequity. Was there a real inequity? Or there was inequity, a simple one letter E and I? I think there's a lot of difference between this. Inequity, we talk of equal opportunity. Inequity, we talk of unkindness. If you look at the UNICEF's data on vaccination, From 2000 to 2019, the DPT coverage has been going up from 80% to 90+%. And UNICEF supplies are majoritarily to the LMICs and LIC countries. So vaccines are reaching. And DPT is also the benchmark for assessment of other vaccine coverages. Relatively, other vaccines also had been supplied to these countries, and the data shows that. There came pandemic, we had a lopsidedness. Post-pandemic, again the vaccine supplies ramped up and we achieved pre-pandemic levels. So what does this tell us? Basically, the issue was during pandemic. During pandemic, many of those wealthy countries received the vaccine, and as we saw in one of the slides here by, I think, Dr. Christopher, Africa was blank. I think it is not inequity, it's inequity, I guess. I think we are addressing here the issue of collaboration, the issue of local manufacturing, not for just other vaccines, to be prepared for the future pandemics. For that purpose, I guess we need to be sustainable. And that is where the issue of collaboration comes in, the discussion on collaboration comes in. In fact, if you look at local manufacturing, the thoughts on local manufacturing has come a full cycle. Several decades ago, I'm sure even in South America, or at least I know in Asia and Africa, the non-profit institutes or state-owned enterprises were the ones who were manufacturing the vaccines. And over a period of time, several of them disappeared and could not sustain, probably because of increased requirement of GMP, QMS, and most importantly, investments on the complete product life cycle, which was not possible by these institutes. And today we are talking of almost 40 to 50 projects, new projects, local manufacturing, around 30 plus are in Africa itself. It is very important to have these local manufacturing from the point of view of ramping up capacities to be prepared for a future pandemic, and therefore today's sustainability. So when this has to happen, what kind of collaboration has to happen is very important. The reason I'm saying this is, in my experience, I've been traveling to several countries. Many country governments normally talk of, complying to the captive requirements. Just to give in a very simple way, pentavalent is procured by the countries. I'm talking of the low-income countries. Pentavalent is procured, tetanus toxoid is procured, hepatitis B is procured. When we discuss with the ministry agencies, they say, we would like to have a facility here. Since we are already procuring this, we will manufacture these products. I don't think that is important. That will not give the sustainability. What is important is to bring in vaccines where, the reason I'm saying this is, there is a conundrum here. If we bring in these kind of regular vaccines, that is, older vaccines into manufacturing with the local entities, you will see these are already licensed and large capacity manufacturers are already there. Their pricing will go up. That is because of the volumes being, the capacities being vacant there, and also the overhead cost allocation goes up. Now, when you look at newer vaccine, the conundrum is that if we seed these projects with new vaccines, the product rolls out after 10 to 12 years. And these new facilities cannot sustain for so long to generate revenue. So we need to find a via media here. Obviously, another issue here I just want to digress a bit and talk of. Unfortunately or fortunately, we have been having solutions and scampering for needs. The Western world or the North, if you look at excellent epidemiologic data available, and what they do is they look for solution in the form of identifying vaccines, because they know the need. So need drives the solution. And these solutions are thrust upon us, we scamper for the need. Product is available, we look for need. I think we need to have a disruption here. We have excellent intellectual capital available in all over emerging world. The issue is that we are kind of getting used to the donor-recipient activity. The Western world has set up laboratories, infrastructure available, and our intellectual capitals are normally towards compliance mode. Yes, we need this. Yes, we can offer this. I think we need to disrupt here, and that is the way forward. in becoming self-reliant. The way we disrupt this, we need to have our own epidemiological data. Probably that will help us look at some of these existing manufacturers who have the newer vaccines. They are able to provide this based on the epidemiological data to those regions, because the existing vaccine manufacturers who have ramped up the conventional vaccines, as I said, will create a lopsidedness. When we look at newer vaccines, I will take an example of our own institution. We manufacture the typhoid conjugate vaccine. We are the innovators. In fact, I should share here with you that the first innovator, I think to my knowledge, an innovative vaccine came from the developing world, the conjugate typhoid vaccine, and it was introduced the first time, correct me, Mr. Suri, it was introduced in the Indian pharmacopoeia. Normally, we are known to introduce all vaccines complying to the British pharmacopoeia. The first vaccine, which was first in the monograph, came as, it came into the monograph as an Indian pharmacopoeia monograph, rather. So when we introduced, WHO did not even look at prioritizing this vaccine. There was no such recommendation. We had to do this, and when we talk to countries, we look at, we hear from them that yes, there is endemic situation for typhoid, but then there is treatment. So this is an optional vaccine. But then we went ahead and doing this. The simple reason why I'm sharing this thing is that there are two issues. One is innovation, maverickness. The other one is, if we had epidemiological data with us, possibly there would have been several countries would be picking up the vaccine. It took 5 years for us to even fulfill the funding available for this vaccine. So while it is very important and critical that local manufacturers have to be supported for ramping up capacity, we have to also look at balancing the sustainability and affordable pricing. And affordable pricing is another big chapter we could discuss on and on. And also in terms of product portfolio. And this is important. As the other speakers were mentioning, during COVID, as Mr. Suri was mentioning in his presentation, during COVID, there were interesting collaborations that we saw. Innovators versus manufacturers. And I think this model can provide some solution to future in terms of collaboration. And talking of collaboration, I think this is not new to us. I should share with you, in 1975, India stopped manufacturing of drug substance of polio. There are 4 or 5 fillers ever since. With 1.4 billion population, with 26 million birth cohort. The birth cohort of India is more than the population of several countries. Just fillers. We only formulate fill, and today India has eradicated polio. Excellent collaboration. The bulk comes from another country, Indonesia. As I'm speaking today, there was another collaboration that is happening in the South America is for an influenza vaccine. A Russian company is supplying to a small country in South America the influenza vaccine. It was a big debatable issue. I have been reading this. There was a lot of debate on whether this will be a successful project. Today I can share that this country is supplying this vaccine to 11 other countries in the whole region. I think this is a success of collaboration, I guess. So it is important to collaborate, and how we collaborate is important, looking at the other parameters that are important. Thank you.
Thank you very much, Dr. Venkat. Excellent. Dr. Andrew, a question also about collaboration. As Industry Association of Developing Countries Vaccine Manufacturers, what role can DCVMN play in fostering collaboration among member companies to work collectively in preparation for the next pandemic?
Thank you, Professor Ruma, and also thank you for the ISI organizer to offer our opportunity here to participate. So as developing country vaccine manufacturer networks, as Dr. Suri already introduced to you, it is a well-established platforms for companies to work together. And as a member companies, as 45 member companies as of today, and 11 companies from China. So we do member companies we've been working together through this network. I think pre-COVID pandemic, it's mainly is about marketing access, market access, and to work on finished product. But after, during the COVID, I think everybody experiences, especially like as a Chinese company like us, we experienced 3 years lockdown. So global supply chain disruption, that was a big issues. And during the COVID, I've been travel to different countries. I heard loud from the government side, from industry side, consumer side, everyone wanted to have the vaccine now made locally. So local manufacture become a trend. And you can see now there's a company, member companies, we work with member companies, with non-member companies, and doing more and more projects, like Serum Institute did, and with Vaxera, like Vax did with our Indonesia partner, Etana, for establishing local manufacturer capability. So DCVMN is really a wonderful platform for member companies to work together within the network and also with out of their work. So that would be the number one. And also DCVMN also foster collaborations by offer opportunity to training the member companies to enabling such as local technology transfer opportunities. The workshop being offered, I think, annually, like in Helmholtz Institute in Singapore, and also in the International Vaccine Institute in South Korea. So those are the opportunity. And last and not the least, it is a DCVMN online training like modules, and do offer member company access to all those, the accumulation of knowledges and also the prior experiences to enable our member companies to work better, work smarter. So certainly this network is do benefit tremendously to member companies and also benefit the global community, global South, global North, because our China is belong to the global North, but we've been working very closely with global South companies in all over the world. Thank you.
Thank you very much, Dr. Andrew. Now a question to Fernando Lobos. Fernando, being Synergium a member of the mRNA tech transfer program established by WHO and Medicine Patent Pool, which are the strengths that this initiative has to better support the regional production and supply of products in the global South?
Thank you, Mauricio. Thank you for the invitation of being here. I'm from Argentina, so I play like local. I'm very happy to be here. So for those that are not familiar with the program, the mRNA program was set by WHO and MPP after the success of the mRNA COVID vaccines, to try to develop the technology and to share the technology with 15 companies around the world. So the technology is being developed by a company in Africa, in South Africa, Afrigen, and from there is being shared to all the other 14 members. So it's a unique program that with the technical support of WHO and MPP is developing this technology not only for COVID but for many other diseases. So the platform is being set, and many of us mentioned today that the collaboration is key. So the new innovation is collaboration, and I think this is a great example of that, because there are 15 companies all sharing the same development and experiences and putting hands-on on that, and we are learning from the others, not only for the good things, but also the things that are going bad. Of course, there are huge challenges to go through, and IP issues and a lot of technology issues. But at the same time, we are putting in the platform new technologies and new and new things that we are discovering. So I think that, of course, it will take a long term to have products from these platforms, but we are sharing the risk. We are reducing the risk because we are moving faster and sharing knowledge, and at the end we will have some products that we can share between regions. So the other thing that I want to mention regarding collaboration is how collaboration is made. I think we need to put some brain on collaboration and decide what you are putting on the table for collaboration to others and what you are expecting from others through that collaboration. So in that sense, many of these companies are in very different regions. We are in Latin America, but others are in Asia or in Africa, etc. So the environment and the situation and the culture is very different. So we need to understand what we want and what we can get from there. And here in the Americas, we have Fiocruz and Bio-Manguinhos and Synergium, both companies leading this project for the Americas, and probably in the future with the possibility of sharing also this knowledge to the rest of the companies in the region. So finally, I think it's a great program. It still needs to demonstrate a lot, but it's one of the unique opportunities or initiatives that we have 15 companies sharing a technology and developing a technology from scratch, and I think we will see the results faster sooner than later.
Thank you, Fernando. Thank you very much. We are partners in this program, yeah? Mr. Suri, although you have made that very interesting presentation, instructive presentation, I have also a question for you. So if you can briefly answer us, as a CEO of DCVMN, what's your vision of regional vaccine manufacturing, and what kind of opportunities do you foresee going forward?
Yeah, thank you. Thank you very much, Mauricio, for this question. I am very sensitive about the time. I think it's lunchtime, so the trickle has already started. So I'll be very short, very brief, so that I don't stand between you and lunch. From the DCVMN perspective, when we are looking at the whole scenario, the landscape of vaccine manufacturers, I think there are three important cardinal principles which we have to keep in mind. Venkat was talking about sustainability. So the first thing that I think everybody has to look at is the sustainability of vaccine manufacturing. It is not proliferation of manufacturing that is required, which was very wrongly being understood by many countries and many people around many agencies, that you should have more and more number of vaccine manufacturers to ensure equitable access to vaccines. No, it is wrong. So what is really required is that you should have sustainable manufacturing, even if when we are talking about Latin America, we are talking about Africa, where inequities of vaccines were reported during COVID-19. I think what is important is a holistic mapping of the whole continent, that what is being manufactured, where it is being manufactured, and how much can ensure that the vaccine manufacturers, whosoever are they are having a sustainable business over a long period of time. That is number one. Number two, which is very important and was missed out, I think, in the first 2 years, was that it has to be an exact matching of drug substance and drug product. What we did was a rat race. We simply went on building capacities for the drug product. And I was told at one point of time when I was delivering a talk in Africa that we have a capacity of 5 billion doses of vaccines. So I said, if you have 5 billion, then why are you asking for something more? It is good enough. But this 5 billion is translated into drug product, not drug substance. Drug substance is 0. So then if a pandemic strikes, what will happen? Again, you'll be back to square one. So I think this is what is important, and this the deeper insights of the people who are working at Africa CDC and PAVM. They have together worked out a plan to build the equal capacities for a drug substance, and progress has been made in at least the last 1.5 years. So that is the second one. And the third aspect is in terms of, we have been talking about South-South collaborations. So it is a cultural compatibility which is important, which we need to understand. You know, you are not Americans, you are not Europeans, you will not be able to breathe the way they breathe. So you will have to understand what are your limitations, what are your problems, challenges, whether it is power, whether it is floods, whether it is anything else. So we need to speak the same language, we need to understand the same issues that the other people are facing, and then start working together as a closely knit family to deliver what is really required. Thank you, Mauricio.
Thank you very much, Mr. Suri. It's very important what you said. And I'm afraid we will not have time for any more questions and any questions from the audience. We need to close our panel now. I want to thank you all our guests. I ask you to give an applause for all of them. And thank you for helping to make our symposium stronger and better. Okay? Tomorrow we will have our board meeting to continue discussing our issues, our strategies, and trying to make our network stronger. Thank you very much. Thank you. And now, hora do almoço agora para todo mundo.
Obrigada, senhor Mauricio Zuma. Thank you for all the presentation. Thank you. Gente, só minutinho, mais minutinho.
Just a second, just one more second. It's very important information. And we would like you to pay close attention to what I'm saying. You know the situation that Rio Grande do Sul is going through, the problems that Bio-Manguinhos is asking tomorrow. Obviously, we're pressed for time today, but if you could bring water or a basic foodstuffs or toilet paper and food for pets, for animals, for example, for household animals. If you could bring food and leave the food over here at the entrance for donations for Rio Grande do Sul. We'll be back in 1 hour.